today I will talk about defective X-gating in the potassium channel TASK-1 and how this channelopathy connects TASK-1 with sleep apnea. I talk about work. This is mostly published, and in October finally we got published. And as you all know, there are many different ion channels in the human body, and most of them are structurally related. The biggest class are the potassium selective ion channels, and in the TACA group, our research was focused on the K2P channels, the two pore domain channels. In potassium channels, you usually have 4 subunits coming together forming one functional pore, whereas in K2P channels, only 2 subunits are needed to form a functional channel. And K2P channels are expressed in almost every cell in the human body. And they play a very important role in controlling the resting membrane potential. First, they were described as leak channels by the original Hodgkin-Huxley model. But this is actually not true. We have many regulators for these channels. We have physical and chemical stimuli and cell signaling pathways, pH, and many more regulators. And then these channels are expressed in so many different tissues in the human bodies, and they play such an important role for keeping excitable cells hyperpolarized underneath the firing rate. A malfunction of these channels can lead to a number of disorders and diseases. In my talk, I want to focus on 2 channels, which is TASK-1 and TASK-3. A loss of function in TASK-3 leads to the severe developmental disorder called Berg-Barrow syndrome, and a loss of function in TASK-1 leads to pulmonary arterial hypertension. Also, TASK-1 is implicated in sleep apnea. It's a very common disease. Up to 1 billion people worldwide suffer from obstructive or central sleep apnea. It occurs when breathing is repetitively stopped during sleep. With central sleep apnea, there's a problem with the sensing of the oxygen and the signaling of the oxygen level to the brain, whereas in obstructive sleep apnea, we have a physical blockage of the upper airways. However, the diagnosis is not always very distinct, and many forms like complex sleep apnea, mixed forms also exist. And both forms are usually treated with nocturnal oxygen. And there is an implication that TASK-1 plays an important role in sleep apnea. Because it is expressed in the chemosensitive regions of the brain, it plays a very important role in the carotid body, where it's responsible for the oxygen sensing. And it's also mechanically involved in breathing, like in the vascular smooth muscle cells or the hypoglossal motor neurons. And all of this makes TASK-1 an interesting target for the treatment of sleep apnea. And indeed, there are currently clinical trials, the so-called KOALA trial, where inhibitors for TASK-1 are tested for obstructive sleep apnea. However, this evidence is mostly circumstantial, and there is no clear mechanistic link between TASK-1 and sleep apnea. However, our colleagues from the Deciphering Developmental Disorder study were interested in the connection between de novo mutations and resulting developmental delay. And they identified 28 novel genes which are very prone to de novo mutations and cause the developmental delay. And one of these genes was KCNK3, which encodes for the potassium channel TASK-1. They informed us that all probants with a missense mutation in TASK-1 also had sleep apnea. And we then investigated this further and found in total 9 probants, each heterozygous for 1 out of 6 separate mutations in KCNK3. And they all shared a common phenotype with developmental delay in sleep apnea, and therefore we term this disease developmental delay with sleep apnea, or short DDSA. And this is the overview over the data where you see the phenotype of the patients. I want to highlight two aspects. First of all of our patients had sleep apnea, they had an increased apnea-hypopnea index, and they needed nocturnal oxygen. The other thing which is interesting is in blue, you can see the mutations which are sitting in the middle of the protein. They seem to have a much more severe phenotype compared to the patients which carry a mutation towards the C-terminus of the protein. And the patients who have a mutation in the middle of the protein also show an abnormality of the limbs. And here's a brief overview over TASK-1 channel. This is a crystal structure in which our lab was involved. And it is important to mention for the talk that not only two TASK-1 subunits can form a functional channel, but also TASK-1 and TASK-3 can form a functional channel together. The key elements of TASK-1 are the cap domain, the selectivity filter, here with 4 potassium ions bound in it, and the so-called lower X-gate. It's like forming X. And when we look at the DDS mutations, and we find that they're all located in the M2 or in the M4 helix. And interestingly, when we look at them in the crystal structure, we found that they all cluster around the X-gate. We did electrophysiological recordings to study these mutations, and we did 2 electrode voltage clamps, excise patches, and single-channel recordings. And here you can see the TVC recordings, the wholesale currents. And you see that all of these mutations have increased wholesale currents compared to the wild type, with the only exception is the L239P mutation. And since our patients are all heterozygous, we tested also the co-expression with the wild type. In black, you see the homomeric mutant channels, in white, the heterozygous channels, and they're both considerably bigger than the wild type current. The only exception, again, the L239P mutation. And we wanted to understand, why do we see bigger currents? Is it because we have more channels expressed in the membrane, or do the channels have a greater activity? And our first trial was the co-expression with TASK-3. Because from pulmonary arterial hypertension, we know that TASK-3 can help the surface expression. And under this condition, we find that all of these channels have a greater activity, including the L239P mutation, which suggests that the channels have a greater activity. To investigate this further, we performed single-channel recordings. This is the zero current level. The flickerings and bursts are opening events, and you already see in the raw data that the mutant channel open much more frequently compared to the wild type. And indeed, the analysis show that we have a 10 to 20 times higher open probability, whereas the single-channel conductance is comparable to the single-channel conductance of the wild type. So we wanted to understand, why do we have this higher open probability. Therefore, we performed dynamic simulations based on the crystal structure in the closed state. In the wild type, we performed runs for 500 nanoseconds, and we did not see an opening event. However, inserting the N133S mutation into the M2 helix shows an opening event in 3 out of 3 runs of the X-gate. This area opens. And the same applies when inserting the L239P mutation into the M4 helix. So we now know that the DDS mutation caused the opening of the X-gate. To understand why this is happening, we were looking again into the crystal structure, and it is known that the alanine, which sits directly in the X-gate, mutations here cause a higher activity of the protein. And now we found the N133S as a position for our mutations, also causing a greater activity. And we think that this hydrogen bond is very crucial for keeping the X-gate in the closed state, because inserting any other amino acid in this position causes greater activity. And we have two gates in TASK-1. We have the X-gate, and of course we have the selectivity filter. We wanted to understand if these mutations also interfere with the selectivity filter, and therefore we performed pH experiments, and we found that the mutants, like the wild type, are activated at high pH and inhibited at low pH, and this is true for all the mutations. As a control, we also mutated the pH sensor, and we found that these mutations only affect the X-gate but not the filter gate. Another way of regulating task channels is GPCR mediation. The activation of G-alpha-Q leads to depletion of phospholipase C, which increases the level of DIG and inhibits the task channel directly. And we know from previous studies that mutations directly in the X-gate abolish these regulation. So we wanted to understand if our mutations, which cluster around the X-gate, have a similar effect. And therefore we performed experiments with carbachol, because carbachol activates endogenous muscarinic receptors in oocytes. And here you can see how adding carbachol decreases the wild-type TASK-1 wild-type currents, whereas in the under heterozygous condition, adding carbachol here, we don't see a decrease or almost no decrease. And the same is true for all of our mutations, even under heterozygous conditions, which mimics our patient condition. And I would like to highlight that under these conditions, also the L239P mutation is twice as big after adding carbachol, because it cannot be inhibited, and here we see it's a gain of function. And to not rely only on endogenous receptors, we also did the co-expression with the P2Y receptor, which can be activated with ATP. Again, wild type and P2Y together co-expressed. Adding ATP reduces the current. Under heterozygous conditions, we don't see a big decrease anymore. And since TASK-1 and TASK-3 can also form functional channels together, we also tested this condition, and apparently the TASK-1 is dominant here, and we don't find any, we found only very little regulation by GPCR. Other physiological regulators are diacylglycerol, which inhibits TASK-1 directly, and we see the dose-response curve is shifted compared to the wild type. Only at concentrations which are not physiological anymore, we see a response. And the same applies for anandamide, another physiological regulator. So we do have these overactive channels, which causes sleep apnea and a developmental delay. And we wanted to understand if there are inhibitors, because we know there are a lot of TASK-1 inhibitors. And we tested the ones we are using in the lab, but also we tested drugs which are in clinical use. And we found that they're all very responsive still to our DDS mutations. And the one which was working the best is what we call the Bayer blocker. And it's a drug with a very, very high affinity. Here you can see the crystal structure with a Bayer blocker bound directly under the selectivity filter, so quite far away from the X-gate. And in red, you can see the dose-response curve for the wild type, and we have an affinity of 1 nanomolar, an efficacy of 1 nanomolar. And even though the dose response curves are shifted for the mutant channels, they are still reactive in a nanomolar range. And I mentioned before that there are currently drugs in a clinical trial for sleep apnea, and a very similar one compared to this one is used for these trials. So this brings me to the summary of this story. In healthy cells, we have small potassium outward currents by TASK-1, which can be easily inhibited by GPCR inhibition. However, in our patients, we have much bigger currents, even under heterozygous conditions. And these difference comes even more pronounced when we look at GPCR inhibition, because here the currents are even much bigger. And with a constant outflow of potassium ions, the cell can't depolarize anymore. But these channels, these mutant channels, are still subject to drugs. They're trackable, and we can do something about it. Regarding our patients, we can't help them with a developmental delay, but we can possibly treat their sleep apnea and therefore improve their quality of life. And we also hope that our research contributes in general to the understanding of sleep apnea. I want to thank everyone involved in this project. I had a really great time in the TACA lab during my postdoc. The Baukrowitz lab hosted me when the Oxford University was closed during the pandemic and also contributed to the project. I want to thank Thomas Müller from Bayer, who made the drug acceptable, our colleagues from the Deciphering Developmental Disorder Study, Carolyn Wright and Matthew Hurls, and the clinicians who collected the patient's data. Thank you.
Very nice talk. Congratulations. One sort of technical question. Do the overactive mutants damage the cells that you express them? The reason why I'm asking is because your heterozygous had essentially the same amplitude as the homozygous, right? Plus, it seemed that the effect on open probability was higher than the effect on the overall amplitude. So I'm just wondering, if you kill some of the cells, you kind of select for the lower expressing ones. Can it explain these results?
Yes. I had to play around with how much RNA I was injecting, and I also tried different expression times to balance it. For the DDS mutations, I found quite suitable times to compare it. But I also had the N133S mutation, where I mutated into a glutamate, and this killed almost all of the oocytes, so it was very difficult to record from this. And a lot of controls here also resulted in a lot of dead oocytes. So yeah, these are very strong mutations which don't make oocytes happy.
Yeah. So really nice talk. So with the L2P mutation, you have an increase in the open probability, but the whole cell current is the same, suggesting either a decrease in trafficking or conductance. Do you know which?
Oh, no. The thing is, we are looking at oocytes. So we know that we know we have higher open probability. We know we have a similar single channel conductance. So to have a similar whole cell current, we have to have a decreased surface expression of the mutations. This is what's happening. And these studies are done in oocytes. The only thing which we can really resample with humans is the single channel recordings, because these are properties which are within the channel. And I want to say that it's about the surface expression, and to really understand what's happening with the patients, we would need to do a biopsy to understand if the surface expression is also decreased here.